Thank you all for the exciting talks. And let me begin the question with Aubrey because he gave the keynote for this session. So during your talk, Aubrey, you did mention the two words rejuvenation and regeneration. You distinguish between the two. Could you define that for us, please?
Yeah, sure. I probably should have defined it a bit more carefully when I was speaking. So regenerative medicine, regeneration, is, to me, the recreation of structure that has been damaged. And it could have been damaged by anything, like by trauma, for example. We heard, of course, yesterday from the sister of a boxer who was badly injured a long time ago. Regenerative medicine exists partly to help people who have suffered damage that has nothing to do with aging. But since I think of aging as the gradual accumulation of damage, I think it's reasonable also to regard rejuvenation, in other words, the restoration of a more youthful state, a state more like a young adult, as regeneration, as a type of regeneration or an application of regeneration. Now, there are aspects of rejuvenation that would not normally come under the heading of regenerative medicine as it's traditionally understood, because they're not necessarily to do with stem cells or tissue engineering. We've heard about some of those today, of course. For example, the removal of senescent cells. So really, I think of rejuvenation as regenerative medicine and other medicine applied to aging, but a large proportion of it is regenerative medicine applied to aging.
Thank you. The rest of the panelists could participate. Professor Dixit, do you have any comments on that? And even the audience can pitch in, because this is just to promote discussions amongst all the participants here.
I must, sorry, I just want to let you know that I think also, Mike, that I can barely understand because I think there is some sort of echo that comes, so I can't really understand.
The question was the difference between rejuvenation and regeneration. So do you have it? Aubrey explained his point of view. I wanted to know, since you also work in the same field of inflammation and rejuvenation, I wanted to know what's your opinion on that.
I think those are, it's kind of a philosophical question. Rejuvenation is of something that was there and we restore it back. And, you know, for example, organ like thymus or any other organ for that matter, we bring it back to whatever is the baseline. Regeneration is typically a developmental term, and I don't know if a limb is cut, that would be regeneration. I don't think we can do that in humans. So I don't know, maybe the stem cell biologists can comment more upon it. I'm just a simple immunologist.
Okay. Thanks. Aubrey, you are embarking on this combination therapy or experiments, not therapies, but experiments. This is 1,000 mice experiments. How does the field benefit? Of course, the field would benefit by looking at what combinations work, what combinations don't work. But apart from that, do you have any plans specifically for data sharing amongst the community? What are the plans?
So, yeah, let me answer the last part of that first. We're in a very privileged position at LED Foundation that our funding comes entirely from philanthropy. We don't have any people who are working for us as a way of getting publications so that they can get their next job. People who work for me don't go off and work somewhere else later. So the main thing is that we can be very public about what we're doing while we're doing it, rather than worrying about priority of publication or protection of intellectual property or whatever. And that means that I'm actually putting updates out on social media every few weeks about how the study's going. We're also, of course, posting those updates on our website. But yeah, it's a very privileged position to be in. I think that what the field's going to learn actually comes in multiple forms. Certainly anything that works, obviously that's going to come out with increasing statistical significance as we move forward. I'm certainly hoping that some of our combinations will be showing at least a trend towards efficacy by the end of this year. That's less than 1 year after the experiment began. But certainly, even though the experiment will not be complete for maybe 3 years. Of course, we don't know any more than one does in any mouse experiment whether the interventions that work will translate successfully to the human setting. But I personally believe that regenerative and damage repair modalities have intrinsically a better chance of translation to humans than what I like to call messing with metabolism, small molecule pharmaceuticals. On top of that, there is, of course, the rhetorical value of an experiment like this. It was nearly 20 years ago that I first coined the term robust mouse rejuvenation, and I defined it essentially as take middle-aged mice and double their remaining lifespan, which is the exact same milestone that I'm talking about hoping to achieve with this experiment. If we don't, we'll try another bunch of interventions, and so on. But the reason I coined that term 20 years ago, and the reason why I feel that experiments on mice are really valuable in the context of aging, whether or not they end up translating, is because when you can take something that has more or less the same body plan as you and me, it has legs and fur and stuff, and you double its remaining lifespan starting from an age when it wasn't yet sick, but it was in middle age, that's a lot more interesting to the general public in a visceral kind of sense than if you do it to something with wings, a fruit fly or a nematode. And as such, I believe that this is the kind of experiment that can really move the needle in society and in the corridor of power to get more, to really launch a proper bona fide war on aging, which I've been trying to do for rather a long time.
The question I was, hello. The question I was asking is, do you plan to create a database, a searchable source of data? I understand that you want to put it on the social media, you also want to put it on your website. Given that there are 1,000 mice that you're actually experimenting with, that's a lot of data to handle on those two portals. So maybe putting them on, putting everything in a very structured manner on a database, a searchable mode would be more helpful for the community.
Okay, thank you. So of course, as I mentioned in my talk, we're not just measuring how long these mice live, we're measuring a whole bunch of aspects of health. The survival aspect, the lifespans, we're certainly going to be keeping updated in a properly quantified way, but that's just Kaplan-Meier curves. That's not very difficult. The difficult part is all of the data we're gathering on blood markers and functional characteristics like grip strength and gait and treadmill and memory and so on. These things are certainly quantifiable as well, and we're gathering that data in a quantified way. And we are right now actually building the database in which to store and contain all that stuff. So for sure we'll be making all of that data available as well. But Jesse, you're completely right. It's a lot of data, and it's a big challenge just to structure the data, let alone to display it. So we are probably not going to be in a position to make that that leap for another few months anyway.
Thank you. To expand on that topic, Dr. Dixit brought the problem up earlier. We, in the clinical end of things, we need to identify better biomarkers of biological aging in humans that even consider the fact that different organs may be aging at different rates, depending on the insults on the specific organs. So any insights, suggestions? I get it. There's confusion in the field on what we could go after, it seems to me. And any suggestions on something that you think is robust, simple,
Yeah, no, I don't really have any magic bullet suggestions there. At this point, of course, everyone's very excited about the rise of epigenetic clocks and other omic clocks, and I certainly will be interested to see how much more progress can be made in, in improving their predictive value. It may be that we're already hitting a point of diminishing returns there, just because, as you say, different organs age at different rates and so on. So we'll have to see. But at the moment, I think it's fair to say that the most reliable markers are the traditional ones, the ones that measure function and changes in the things in the composition of your blood and so on. I think we're getting further with that.
I can add to that a little bit.
Yes.
One big problem is that when we're looking for biological clocks, we're looking at tissues, different tissue, but many tissue have a different regeneration rate. So there are some tissue which accumulate older cells a lot more than others, right? And that makes it more complicated. And that's why a lot of these talks don't really talk to each other that well.
Yeah, there are a lot of projects that are ongoing with regards to biomarkers. For example, Harvard, Vadim Gladyshev has a project wherein he is integrating a lot of different professors from different universities and groups together to create a whole database on biomarkers that have been tested and those probabilistic biomarkers. And also there is, Tina Woods was here a little while ago, maybe you should speak to her. She's compiling a biomarker project for the Foresight Institute in the Bay Area. So there are a lot of initiatives that are ongoing as far as the biomarker efforts are concerned, not just the epigenetic clock that exists. Professor Dixit, were you able to hear this? Do you have any inputs on this?
My apologies, I couldn't hear anything.
I'm so sorry.
I can only hear you.
Okay. So how do we fix that? Is it possible? Oh, okay.
It's the computer,
Yeah.
closer to the computer, that's why.
No, but then it's important for him to hear everybody else.
That's why he's not able to hear anybody else,
Yeah,
because you're
okay.
closest to the computer.
I'm so sorry about that. Yeah. The next question I have is for Shi. I'm really very excited about your project. What's next for Xtherma? I know you talked about a lot of different goals and a lot of different collaborative efforts. What's next? I know you talked about 120 days, right? Or hours?
Hours.
Hours, okay.
Yes.
So what's next?
So yeah, thank you. It's a really exciting time for us. So biopreservation traditionally has been largely overlooked since 1950. So we have now seen major breakthroughs. So we're here to make the impossible possible, and now through the small animal data, and now we get large animal data, and we got FDA's recognition for breakthrough designation. So now we have a full force going forward, and then engage with the clinical trials, and then get this to the market to start save patients' lives through the kidney transplant. Yeah, that's one of the goals. Yeah, on the other side, actually for supporting the biomanufacturing goals nationwide for biotech and biomanufacturing, harnessing this entire supply chains. Xtherma is fully geared up with our partners, actually to bring more robust biopreservation logistic management for the customers. So that's the two main goals for us. Thank you.
Fantastic. When you look at your data, do you see any organ specificity or cell specificity in this period for which it survives outside of the body?
Yeah, actually, that's the thing we're very excited about. So, we have tested multiple different organ systems. We first test on the mouse heart. It's a small heart, but it's a full functional heart. So we demonstrated 6 times or longer extension time compared to conventional state-of-the-art method. And then we jump into the entire red penile tissues. So, our leading transplant surgeon actually downworld first the entire male genitalia organ transplant. So we tested on the rat system and demonstrate 72 hours in success for retransplant in the control group, complete necrosis. And then we move on to pig kidneys. So we think so far we have not seen like this is organ specific because actually peptoid is really here to interact with water, interact with ice, but we don't think they have like a biological functions in this case. So give us a great hope it can be like organ agnostic. Thank you.
Sorry.
Yeah, I'm not going to ask the question that Evan was asking me. So we've talked about rejuvenation, making things how they used to be. We've talked about repairing. I would like to dangle an ethical noose and see if anybody's prepared to put their head through it and ask, is any of the data that you're collecting suggest that we could actually make an organ or a tissue that's better than normal?
So I've always felt that the boundary between what's a therapy and what's an enhancement is inherently a very grey boundary. And of course, a lot of people feel that it's even a matter of definition whether rejuvenation back to youth is an enhancement simply because of the side effect that it may let people live longer than anyone's ever lived before, and so as such the readout is an enhancement even if the function at any particular time is simply restoration. It's a rather crazy kind of grey definitional area. But, I mean, if we look at what we're doing even today with the creation of iPS cells, these are cells which are younger than young in some ways. And so I can imagine that we're going to see that again and again. We're going to see the creation of therapies that reduce biological age to something that is less than that of a young adult, while nevertheless, of course, maintaining adult physiology. And so I guess that could be called an enhancement.
I have a question in turn for you. Yeah, sure. Do you want to give it a go? See?
For me, I think regeneration is really, we're talking about regenerative medicine, so the medicine for the current definitions for getting a cure or have a medical need, so we're solving a problem. We'll say rejuvenation is really regeneration is about to live and rejuvenation is about to live better. So that's how I think about this.
Okay, so Amit.
Yeah, I mean, the cars we are making are definitely better than regular normal NK cells. I mean, these cars that we're making are better than normal NK cells. They are rejuvenated. Their natural cytotoxic function is improved, and now we've added another arm to it, which increases their specificity even more. So yeah, I mean, we're making cells which are better than youthful cells too.
So I hope I get to ask you the question. Thank you. So my question is, when you say better, what do you mean by that? Are you talking about at the whole organism level, function, structure, as well as phenotypic? But you asked about organ. So at the organ level, again, you have to answer whether you mean structural improvement or resetting of the clock from a structural perspective or from a functional perspective.
Wait a minute, Graham.
So, from a functional readout, what I thought one of the panel would have answered was that if we could improve the hematopoietic system, then perhaps we'll have a more effective athlete. There are aspects of how our lungs function. Pardon? Oh, well, that's just a different problem. That's why I made it specifically to do with an organ or a tissue. You then have to work out how you can make the car still work on the road with a bigger engine in it.
Evan, do you have a question?
Oh, Evan definitely has a question. He definitely has a question. I just don't think he's going to ask it.
Without Evan asking a question. Sure.
This is really going back to, I think basic science, right? So we've seen in the laboratory flies that live longer, worms that live longer. In my neighborhood in San Diego, I had a neighbor who had 2 Galapagos turtles, and he was trying to give them away to somebody who was young because he was old. I see the newts that you people have chopped their arms off and they regenerated. And I always say, and then in New York, there are these clinics where you buy young blood to infuse based on the parabiosis model. And I just make comments, you know, and my thoughts are, well, people think we can live a better life, we can regenerate. I don't know if that's really the way, but it's still based on science, right? So medicine's based on science, I think. Sometimes it seems to be like voodoo, but I'm wondering if we go back to some basic principles. I just asked like the question, the newt. Why does it regenerate? You know, how does it know what to do? And those are questions like, oh, we don't know. And we would love to be able to. If I cut off my little bit of my finger, I could regenerate it really quickly. So Aubrey, I mean, you've looked at all this data and you've been involved in this thing. So what are your thoughts in general? Do you think these animal models are just big waste of time?
Animal models are very far from a waste of time, but you have to use them, you have to know what to look for, really. So, you know, there are definitely species that live a lot longer than humans do, but that's generally because they have, let's call it, a less difficult aging problem than what we have. So, for example, bowhead whales. They have an easier problem because they're bigger, so they have a smaller surface to volume ratio, they can have a lower specific metabolic rate, and that means they have lower oxidative stress. There are organisms that seem to be completely immortal, but they don't have brains, or they have brains that dissolve when they revert back to being a polyp, things like that. So that's not terribly useful, but yet there are other things that are much more useful. Like, for example, one of my favorite things that's currently a big research topic in the field is how naked mole rats avoid cancer so well. They live a ridiculously long time, they have extraordinarily good cancer resistance. It was discovered not long ago that they have a particular variant of high hyaluronic acid in their extracellular matrix that appears to be very important in this. You know, that's the kind of discovery that might well be translatable to humans. So yeah, we have to be intelligent about this. And certainly, I mean, you mentioned the example of salamanders regenerating limbs. This is absolutely a big research topic right now. And in fact, it's tomorrow, isn't it? Ellen's coming? So hang around for Ellen Haber-Katz tomorrow. She'll be talking about an unusually regenerative strain of mice. So yeah, we absolutely need to know these things. Of course, as a technologist, I'm always thinking about how much we need to know in order to be able to manipulate nature in a desired way. I'm not curiosity-driven. I don't find things out for the sake of finding things out. But yes, there are certain areas where we definitely need to know more.
Thank you. So the next question I have is for Xi again. So I did see a lot of interest amongst the audience. People approached you, were talking to you about different ideas. So what does it look like? For example, if let's say I want to work with you on a specific project, what is the IP scenario? What is the funding scenario? How does it look like? What fields interest you? What fields don't?
Yeah, very good question. We're a small company. Yes. So, yeah, we here really see need to solve those bottleneck. You know, this is one of several key elements actually giving us a roadblock to allowing us to truly enable regenerative medicine off the shelf, truly enable us to scale up, scale out the productions. So in general, I'm interested in working with you all, frontier researchers and also the developers in different clinical stage, preclinical to clinical phases. So we actually have built a very robust client system. So we have those early access program and through 2 different types. One is we work with R&D forefront. So we offer a small testing samples and you know what you want to testing and test on the protocols and you can have your first-hand experience with our products. And what we come here is like a very robust system. We go through those steps and actually make sure we're not only providing you a bottle of a solution, actually come with expertise in cryopreservation. So the goal again is like to become your partner, actually support your success, not just like giving you a product and let you run with it. On the other side, we also work with large process development partners, so who has very intensive clinical relevant process development. So we provide much more hands-on support with our expertise in this specific field and GMP grade product like for testing. So overall, on the end, like we arrived into the supply partnership. So we work with the partners like to support your success. So that's how we work for business model.
Thank you. So now I have a couple of questions for Professor Dixit. And now I guess you can hear me? Hello? Hello? Okay, fantastic. So inflammation is multifactorial, and it could be causative in some cases. It could be a result of some of the processes, or when for some of the hallmarks are impacted. So now, is it required at all when you are creating a therapy or intervention strategy to distinguish between the result and the cause? And that's the first question. The second question I'm going to ask both together in the interest of time. How sustainable is this thymic regeneration that you see with your CR mimetics?
I'll ask the second question first. We haven't put up the people that underwent calorie restriction that showed thymic rejuvenation because at that time the IRBs and the Data Safety Monitoring Board did not allow us to follow those people because of HIPAA regulations. But there are new trials that are being designed, and hopefully we'll be able to see if how long this process remains active. The good thing about thymus is that you really do not need to produce a ton of new T cells. And one of the ideas in the field of immunobiology of aging is that even if the thymus produces a limited number of T cells and it remains active only for a small time, that is usually sufficient to expand the T cell repertoire in a way that then you can then go in with specific immunizations or vaccines or other interventions that is beneficial. So you do not need the thymus to be active for a very long period of time, apparently. First question, I think, is again, I think, more of a philosophical question that you're asking, result and the cause. But I think the inflammation of aging is very unique. Obviously, you could compare it, like if you have a, how do I say it? If you have in your kitchen a tap that is, dripping, and that's like the inflammation of aging, and you want a plumber to come in and fix it. Let's say that the macrophages or stromal cells, the plumber has to come in and it has to shut down the whole water supply in the house, right? And that sometimes happens in the context of inflammation with the trade-offs that happen in terms of loss of tissue function or the loss of organ function. So, but the data that we and others have accumulated, what's matter is inflammation is a very broad term, like you mentioned, and within that inflammation, there are very specific mechanisms that alter or perturb homeostasis. So it's important to target those, and the question is not. And the idea is not to kind of get rid of those, but to kind of just lower them enough that that the sustained activation has gone back to kind of normal. And this has at least been shown genetically from the basic science studies that have come out from multiple groups. I think the evidence as far as human interventions is concerned is something that is ongoing. There are studies, for example, from this trial called as the CANTOS trial, which was a trial done to lower or to neutralize IL-1 beta by canakinumab, and this was done to see if that reduces cardiovascular events in people that have inflammation. The trial was successful. It showed that indeed one can lower those events and one can lower inflammation. What unfortunately was not looked into was that these were all older people, and we know that IL-1 beta is higher in elderly. But we don't really know if those older individuals had improvements in other parameters of aging that we can quantifiably measure. So I think the studies in terms of targeting aspects of inflammation required to be done very carefully, especially interventional studies in humans, to see if dialing back some of these mechanisms or specific mechanisms have a real impact on biology of aging and health span.
Thank you. Amit, you had a question for Professor
I have a quick
Dixit.
question regarding the first part of your answer regarding thymus. Are you looking at any functional? I don't even know if you can hear me. Can you hear me? Oh. Oh, can you hear me? Dr. Dixit, can you hear me? Okay. So my question was, is there any plan to look at the T cell diversity, which we know significantly is affected by age, especially in CMV seropositive individuals, 25% of all their CD8 T cells are recognizing very few antigens. So with calorie restriction, if you see improvement in thymic function, at least in terms of ratio of these CD8 T cells with, say, naive T cell population, is that something that you're looking to do at all? Or what do you think is the functional consequence of this regeneration of thymus upon calorie restriction?
Yeah. So first and foremost, let me say this again. When this trial was designed, this was not something we predicted, and we only knew this when the data was unblinded. So which meant that we had absolutely no idea, because the trial was complete, people had gone home, and that's when we found this. So we have absolutely no idea as to what is happening in terms of function, in terms of whether these individuals have better T cell function, better T cell repertoire. Other thing is, these are all healthy people. Okay, healthy people means that they do not have restriction of T cell repertoire diversity, which happens in humans after the humans are close to 70 years old. So I do not expect any change in the T cell repertoire. We did do the sequencing of the T cells, and there was absolutely no difference in the circulating T cells in the blood, because the number of recent thymic emigrants that are produced from the thymus are so minute that, and they do not necessarily go to the blood, they can go to the resident organs, resident non-lymphoid organs as well. And those are not accessible in humans. So the future studies are required to do this, but in animal models, it has been very, very effectively shown that if you expand work from my group and from Richard Boyd and others have shown that if you can reverse aspects of thymic aging or prevent thymic aging, and you produce more new T cells, that expands T cell repertoire to prevent certain infections and improves immune surveillance. Those things are much more challenging to do in human beings, and because they need to be done in individuals that have restricted T cell repertoire, and that is not yet being practical. It's not yet have been done, and clinically, I think it remains a challenge.
Thank you. So we've almost run out of time, but I do have one question. Please keep it as concise as possible, your responses. What does the future hold? I know you're all chipping away at the big problem of aging in your own ways. So what should we expect? What should we look forward to from your respective labs and your respective work? I can start with Amit, because I have a lot of questions written down for you, but I didn't have the time to ask. So go ahead, Amit.
We can talk afterwards. I think this is the best time to work on aging. Right now, we have really good interventions that have been proven in various kinds of models. We have been able to collect human data now. We're making great progress in there. The way I look at this is not even controversial to me. We have people who have gained wisdom all of their lives, and then their bodies start failing them. If we are able to find interventions that gives them 10, 15, 20 more productive years, I think that would be good for these people, and I think it'll be good for the society. I'm very, very optimistic for the future.
Okay, so the question, then maybe you could answer the next question as well. What does an ideal longevity intervention look like for you, from your perspective?
Which also improves health span significantly, and I'm talking about physical function, muscle strength, mental acuity, like everything. It has to actually improve quality of life.
I think like for this entire field, we're all about time, right? So we're really working on to bring you the time you deserve. So first of all, no one should die from waiting for the organs for transplant. No one should be waiting in vain to getting a life-saving regenerative medicine. And the next step, we're talking about we can repair, rejuvenate, regenerate and rejuvenate our bodies so you can live a better life. So that's the hope in the future. And I see this is a critical point. Once we get more time, that means we can combine regenerative medicine and data science and personalized medicine all together. This is like just a singularity event, like we could get enormous potentials ahead of us. Future is bright, and let's work on having more time.
Thank you. Let's get to Professor Dixit, your opinion.
I'm a scientist, I'm not a philosopher. I'll just say that I'm just having a lot of fun studying the basic biology of aging because it's one of the most fundamental mechanisms. And I would just like to just say one more thing. You know, the average lifespan in Roland Park in Baltimore is 82 years. And 5 miles down the road in downtown Baltimore, it's 62 years. 62 years. So I think as you guys think about bigger things in life, please also remember that the reduced lifespan that we have in U.S. in many, many places is not because of advances in aging. It is simply because of disparities. And I think as we move forward, we have to probably think about it. And I'll let the philosophers here, opine on that.
Thank you.
Yeah, I'm not a philosopher either, but I do think it's an extraordinarily fulfilling time to be working in this field. One example that I often use is that even as little as 10 years ago, when you were writing a grant application in this field, you absolutely could not mention anything about doing anything about aging. It was always, trying to understand aging better. This was the era in which I like to describe gerontologists as being rather like seismologists. They understand that the thing they work on is bad for you, but they have no aspiration to stopping it from happening. And that was as little as 10 years ago. And now it's completely the opposite. You can't expect to get funded unless you talk about the translational potential of what you want to work on. So that is a seismic, in fact, change in attitudes within the field. And I think what we can expect to see over the next few years, less than 10 years for sure, is a corresponding 180 in the attitudes towards aging in wider society, an abandonment of this thing that I call the pro-aging trance, where people make excuses for aging and pretend that it's a blessing in disguise. So that's really where I see the future going.
We have one last question from Alan.
Thank you all for really great presentations. Aubrey, what you were just saying about aging and having to put it into requests for funding, where in that process is the distinction between health span and longevity?
So a huge amount is made in the wider world, in the media and so on, of the distinction between health span and lifespan. And indeed, one thing that I rather dislike about the kind of form of words that some of our more senior colleagues use is they kind of pander to that. But in fact, absolutely everybody knows that lifespan is just a side effect of health span, that they go hand in hand, and that any kind of variation, which is often called either compression or expansion of morbidity, is just rounding error compared to the advances that have been made in both health span and lifespan over, let's say, the period since World War II. So therefore, it's a very popular question, it's a very popular topic, but it shouldn't be. It's a non-issue. Lifespan and health span go together.
Thank you all so much. Professor Dixit, thanks for joining us. And we can move on to the next panel. Aubrey, please stay on the...
Oh yeah, that's great. Yeah.